We're going to continue with mitosis. This is another set of models. Again, we have the set where the first model where it is in interphase, so the nucleus is intact, the centrioles, which are the little black dots on this model, are together on the side. The DNA will be in the chromatin phase. This will then enter into, after finishing interphase, which is G1S, G2, it will enter into prophase. With prophase, the centrioles have started moving to opposite corners. The DNA has condensed into chromosomes. Okay? At the end of prophase, the centrioles will be at opposite poles and the mitotic spindle will be formed. We jump quickly into metaphase, where we've got the centrioles at the opposite poles. The mitotic spindle is formed. The sister chromatids are lined up on the metaphase plate, getting ready to divide and be pulled apart. Then we move to anaphase, where they have separated and they're being pulled to the opposite poles. At this point, they are now considered chromosomes because they do not share their centromere. And then into telophase, where we have two different nuclei having formed at opposite poles. The cells are going to go back to being the relaxed phase of chromatin. The cytokinesis is occurring, where the cytoplasm is pinching in to form two cells. And at the end of this, we will have produced two identical daughter cells, which will look the same as the parent cell. In addition to looking at the models for mitosis and the figures in the book, we have a slide to look at. The slide is going to be a slide of a white fish blastula. This is a phase during embryonic development where the cell starts to multiply quickly and reproduce and produce a huge ball of cells called the blastula. Now within this ball of cells, different cells will be in different phases at the same time. So some you will find are in interphase, where the DNA is relaxed and the nucleus is intact. Some will be in prophase when they're partnered up. And you will notice this by the darkening of the color, because these are dyed. The nuclear material has been dyed. So when the DNA is relaxed, it's a lighter pink. When they start to condense and form chromosomes and chromatids, it becomes a darker purple. Some will be metaphase, where they're lined up in the middle. Some you will find in anaphase, as they've pulled apart from each other, and there will be some telophase. This is the general photo. Then to look at the different photos, here is and these pictures are on Blackboard as well, on the Blackboard institutional content. Here is a cell in interphase, and you can see the DNA is light pink. The nuclear envelope is still intact. The plasma membrane is visible. The DNA is in the chromatin phase. Then we have a close-up of a slide in prophase. Here the DNA has started to condense, so it's getting darker. You can really see it in this picture. The nuclear envelope is going to have disappeared. You won't really be able to see the centrioles in that they've moved to the opposite poles very well. The next phase is metaphase. Metaphase, they've lined up in the middle, so you can see a nice dark line of little X's across the center of the cell. Then they will move into anaphase. Anaphase, they're moving apart. These are different early to late anaphase, where you can see the space as they're separating. You can see that they're now chromosomes. The plasma membrane is intact. You can see the mitotic spindle to some extent. And then finally, they will enter into telophase. Again, these will be in various phases, early, late, until we finally have it completely separated. But you can see that they still have the spindle attached to them, so that they are still touching each other. At the end of this phase, they will completely separate through cytokinesis, and they will be now two individual daughter cells.